Hi.
Is hosted by myself and Lily Packler-Shahabi, who's our clinical research practitioner at Imperial. And the aim of Horizons is to present a mixture of clinical and preclinical work, but reasonably close to implementation for techniques, approaches, and treatments that might relatively soon improve care for patients with brain tumours in the UK. We will be recording this session, and the video will be available on YouTube about a week after we finish. So if you missed the session, or if you have to drop off halfway through, you can catch up with the rest of it then, and all of our previous talks then as well. It's also worth saying, as Lily already mentioned in the chat, our next Horizons in Neuro-oncology session in March will feature Helen Bullbeck, who many of you will know from Brains Trust. So today it is my great delight to introduce Dr. Eleonora Peerani, who is Head of Research and Development for Precision Medicine at Pear Bio, who's going to talk to us about some of the work that Pear Bio is doing. As a conflict of interest statement, some of you will know that I am the Chief Medical Officer for Precision Medicine at Pear Bio. I have salary and stock options, but this is more an example of me twisting Ellie's arm because of our relationship at Pear Bio and getting her to give a talk. So I will allow her to introduce herself. There'll be about 40 minutes for the talk and then 15 minutes for questions. Ellie.
Thank you very much. Okay. Can you guys see my screen all right?
Yep.
Yes.
Yeah. Cool. Well, good evening, everyone. Thanks for joining me, and thanks to Matt and Lily for allowing me to speak to you tonight. So yeah, my name is Eleonora Peerani. I'm Director of Precision Medicine R&D at Pear Bio. And prior to Pear Bio, I did my PhD at Barts Cancer Institute, focusing on biomedical engineering and clinical oncology, where I worked a lot in the development of 3D models with translational value. And you'll see that coming up a lot in my talk tonight. So yeah, Pear Bio is a tech bio company. We're a little startup. We're based in London and Boston in the US. And we're really focusing on the development of precision medicine tests that can match patients with the most effective treatments for their cancers. So tonight I'll walk you through the optimization of our platform that we've done in-house, and also talk you through our PEAR-GLIO results. PEAR-GLIO is a clinical trial with CI, Matt being the CI, for the study, and where available, I will put in some of the references to PEAR-GLIO and the clinical data that we've gathered in gliomas for the development of the platform specifically. So yeah, Pear Bio uses computational biology to deliver patient-centered cancer precision medicine. So one of the strengths of Pear Bio is that we have a very large pool sample of patients that we use to build our biobank. And that comprises both patient tissues, patient tumors, blood, and their data. And we leverage this biobank to create what we call 3D immune microtumors, which are ex vivo models. They're proxy for the patient that we can grow in the lab. For each patient, we can create these 3D immune microtumors. We create a lot in parallel, and we can treat these different microtumors with all of the therapies, combination, monotherapies that would be available for that patient in the clinic to receive. We test these therapies, we run our automated computer vision pipeline analysis on them, and we produce a report that we hand back to the oncologist outlining which of the therapies that we tested ex vivo would work for that specific patient and which ones would be ineffective. So as you can imagine, there's a scenario where we predict response to at least one of the approved treatment regimens, in which case, we're hoping to help clinical decision-making by increasing the certainty on treatment selection for an oncologist, for the treating oncologist. The other side of the coin is if we predict that the patient is not going to respond to any of the approved treatments, then we can suggest that patient may be entering clinical trials earlier. And we collect all of this data, both the functional, the phenotypic, and the clinical data of these patients. And we can use the results of the functional test plus the clinical data plus any leftover samples to also develop our own target and drug discovery programs in-house. So these are specifically targeted to those patients that we see have no therapies available to them out there. And so we can also develop drugs to serve these underserved or high unmet need populations. So for tonight's talk, I'll be mainly focusing on the precision medicine side of things, but I know a bit about the drugs as well, so we can touch on those later if there's interest. So our platform is made up of fully human ex vivo models made with primary patient samples with matched immune cells. And we use these to quantify therapy efficacy and therapeutic mechanism of actions by using time-lapse 3D confocal microscopy, proprietary computer vision analysis, and a lot of multi-omics complementary data that we can gather through multi-omic assays. And because this is a generalizable drug agnostic, modality agnostic platform, we can test a variety of different therapies spanning small molecules, antibodies, ADCs, cell-based therapies, as well as more kind of exotic mechanism of actions like virus-based therapies, all in combination or as monotherapies, just as they would be prescribed in a regimen in the clinic. There's a lot of cancers out there that have a traditional, very strong link between biomarker and drug efficacy. For example, in lung cancer, if you have an EGFR mutation, you will get an EGFR targeting treatment like osimertinib, and you have a very high chance of response. We at Pear Bio go after cancers that traditionally have a lack of predictive biomarkers, so where there is no strong correlation between the expression of any biomarker and treatment response, and also cancers where there may be very complex treatment regimens, where our treatment agnostic, modality agnostic model really becomes really powerful. And so the three kind of main indication that we focus on are triple negative breast cancer, kidney cancer, and brain tumors and gliomas. The way the test would integrate in the clinical pathway is a patient would present themselves in a clinic with an advanced cancer. If a diagnostic code needs to be taken for analysis, then it will be taken. In the case of gliomas, we can work directly with the resection, so an additional visit of the patient in the clinic is actually not needed. So we will take a little bit of the patient tumor alongside some of their blood in a routine kind of operation. And any excess tissue that is not used for diagnostic is shipped to us. We will run our Pear Bio test, and within 5-7 days, we can return a report back to the oncologist, the treating oncologist, outlining how the different therapies worked ex vivo on the specific patient sample. And the aim, is to help clinical decision making. So for PEAR-GLIO specifically, this is a trial that funded by Pear Bio. And the aim of this trial is actually just to develop the platform. So it's very much an observational clinical trial. And the primary endpoint is to be able to see differential efficacies of drug ex vivo and to collect both glioma samples, which are hard to come by, and correlate them to the responses that we see in our test to patient outcome. And so far, we have only tested 12 patients, and we're aiming to recruit more, to resume recruitment for this clinical trial around April. As I said, Matt is the CI for this, and really the aim is to see whether we are able to repurpose our precision medicine platform and use it in brain tumors. Alongside our functional assay, we'll be running a variety of different multi-omic tests, so genomic transcriptomics, and the strength of this trial over some of the other trials that we're running, and jointly with Lilly as well, is that we run patient public involvement and engagement, which we found really have strengthened our clinical trial. I think it's really important to get people's, both from the clinician side, but also from the actual patients and carers side, their opinion on the utility and potential clinical pathways in which we could adopt these tests. So I'll now walk you through the actual assay and how it came about and how we developed it. So generally, the workflow is we'll receive the patient sample, we'll log the sample, and we'll extract cells both from the tumor and immune cells from the blood. We stain these different cell subpopulation with different live cell tracking dyes. We grow them in 3D into what we call the 3D immune microtumor. This just helps the cells be more happy. Once they're taken out of the body, primary cells, patient cells, don't do too well in solution. So we recreate their 3D architecture to try and mimic what the tumor microenvironment that they experience in our body is. We characterize all of these microtumors, and then we can start our actual drug dosing. So administering all the drugs that the patient could be receiving in the clinic, and we use imaging to try and figure out which ones worked best on a number of different functional metrics. Any excess tissue we also use for other assays and multi-omic characterization. So starting with the first step, once the sample comes in, we will split this. Some of it will go for a fresh assay, which is the functional assay. Some of it will be biobanked so that we can later go back and test novel therapies, for example, if we identify that this patient may be a good responder for one of our novel drugs that we're developing. And part of the sample will be fixed or snap frozen, which enables us to do both spatial omics, so spatial transcriptomics, spatial proteomics, as well as bulk tissue transcriptomics. And all of this data, so the integration between this multi-omic phenotypic functional data is really the strength of our data set. Doing this over multiple patients, we can start identifying patterns in response, non-responders, and is really our differentiator from other drug developers. For the purpose of this talk, again, we'll be focusing on the fresh assay. And so far, we have tested over nearly 800 patients, spanning 11 indications, and with our lead indications being triple-negative breast cancer, kidney cancer, and more recently, brain cancer as well. Okay, so what does the test actually look like? So practically, we receive a tumor resection and we receive the blood. We'll then isolate what we call PBMCs, so peripheral blood mononucleated cells, which are your immune cells, from the blood. And we'll further isolate CD8s, these are your T cells, and we'll stain T cells and the rest of the PBMCs with two different cell tracking dyes. Historically, I mean, typically your T cells are your effector cells. They're the one actually doing the killing when it comes to testing immunotherapies, for example. So that's why we tend to separate them and stain them with a different color. The platform is fully, can be optimized or changed if there's another cell type of interest that we're interested in tracking. That's not the T cells. After this separation, the cells are reconstituted and activated overnight with CD3. In the meanwhile, we'll take the tumor resection or core needle biopsies, again, depending on the indication, and we'll isolate cells from these. So this is a mixed cell population. You will have all of the cells that were natively in the tissue. So this comprises your cancer cells, your endothelial cells, any tissue-resident immune cells, epithelial cells, you name it. Everything that was in the original tumor is retained through digestion. And then we test, we just assess the viability of the cells that we have extracted and the cell number. So if I can bring your attention to the actual brain, we see that we've consistently kind of hit viabilities of above 80%. And in terms of cell number, we're well above 10 to the 6. So we're in the millions of live cells that we extract for these samples. And this is ideally what you want. You want to extract a lot of cells and a lot of viable cells. If we hone in into the 12 patients that we tested through the PEAR-GLIO trial. This is what the numbers look like. So we're getting a lot of cells with very high viability. And this is great because this is the input material that we'll need for running our assay. After we get our single cells, we'll stain them. And as I mentioned earlier, we need to grow them in 3D, identify what the best conditions for their 3D cell culture are. And in order to do this, we usually do a lot of literature research and identify what extracellular matrix proteins are important for the pathophysiological progression of a specific cancer. In the case of brain tumors, hyaluronic acid, tenascin C, laminin, and fibronectin are 4 very prevalent ECM extracellular matrix proteins in the brain. Therefore, we try and test hydrogels formulations that recapitulate these different components for our 3D cell culture. So these are the patient codes that we have tested, the viability of the cells, and these are some, for example, sample assays that we conduct to validate whether the cells are happy in our 3D cell cultures. An example of an assay that we do is viability, so very simple live dead. We culture the cells in a variety of different gels comprising different amounts of these extracellular matrix proteins, and then we see how happy the cells are after culture. We can also take confocal images and look at the morphology and viability of the cells. After a few days, you can already see that the cells are starting to show these elongated morphologies, which is really good because it means the cells are actually proliferating and differentiating into functional cells. And then on the side, we run a lot of other assays to validate our 3D cell cultures, such as looking at the metabolic activity, the proliferation of the cells, morphology, what dyes may be best for tracking different cell subpopulations. We can look at soluble factors that are indicative of cellular activity, as well as optimizing cell culture media for culture of these. And this is generally the process that we follow when we're optimizing 3D cell culture for a new cancer. And so far, we've tested this on 11 solid primary tumor types. Next, we characterize our 3D co-cultures. So on the PBMC side, so on the immune cell side, we can run flow cytometry. This is very standard practice to just characterize how the different immune cell subtypes are present in the immune cell populations in the blood. When it comes to the microtumor, though, we usually perform immunofluorescence, and we characterized, this is an example from a kidney cancer data set, but we characterize the tumor cell percentage, the epithelial cell content of our cultures, endothelial cells, immune cells, fibroblast. So as you can see, there's a sort of wide spectrum and variety in the composition of samples that we test. And this is, I mean, totally expected because of the heterogeneity that exists between different patients. What's important for us is also to retain those cell types in culture. So here we performed the same analysis that's portrayed here on the left, both on day 0 and day 4 of culture. And we're able to show that in our culture, we're not skewing cell types towards preferentially proliferating and becoming cancer cells, or like if we're losing a specific cell subtype, that could be also problematic. We actually retain all of the complex cell types from isolation all the way through culture in our assay. We have further characterized these with proteomics and transcriptomics approaches. So here you're seeing an example of RNA sequencing that we've performed. And again, this is just enabling us first to understand more about the actual cultures and the cells that we're growing, but also to validate that our model really retains that human is a closed model that we can use that retains transcriptional signatures that would be present in the patient, so that we, again, are not altering the physiology of the patient through culturing. And this kind of wide analysis and multi-omic analysis is across many, many patients, is also how we identified one of the 2 targets that we are currently undergoing drug discovery programs with. So again, having these big data sets and understanding patient biology also allows us to develop our own drugs internally. Nice. So now that we have created this 3D cell cultures that closely mimic what's going on in the patient, we can use them to test drugs and approved therapeutics. And we've tested a variety of different modalities, from your normal small molecules to monoclonal antibodies, all the way to PROTACs and oncolytic viruses, so more exotic therapies. In the case of PEAR-GLIO, this is the panel of drugs that we've been looking at, and it's validating. And then we take these patient samples and we test all of the approved therapeutics. In this case, what you're looking at is dose response, because we're still in the dose optimization phase of this study. But each different color, each different line is a different patient, and we can repeat this across all of the therapies that are approved for GBM. In some cases with some drug, for example, in procarbazine right now, that I'm showing right now, you can start seeing at certain doses, a certain level of separation between the responses of the patients. So I would interpret this, again, this is very early data, preliminary data, but you could imagine that these patients down here are the ones that are responding more to this specific drug. You have a group in the middle that's so-so, and then you have a group of more resistant patients. And running these analyses over a lot of different drugs and a lot of different patients allows us to create more predictive, increasingly predictive models by using the assay. How do we actually read out? What's our actual readout for these cultures is what I'll go into next. So we have a subpopulation of cancer cells stained with yellow and encapsulated into a hydrogel at the bottom here. We'll have our immune cells extracted from the blood stained red and blue, which are in solution, free floating around your microtumor. And over time, these immune cells will attach onto the surface of the hydrogel and will start infiltrating and interacting with the cancer cells. That's how we can, for example, study the effects of immune checkpoint inhibitors. We can add a control or a drug treatment also in the media with the immune cells. And we add a viability dye, which is green, which fluoresces green when the cell is dead. And we can run these microtumors through a confocal microscopy. We take time-lapse or daily images of these cultures. And this is the type of... And we have an automated microscope as well that enables us to do a higher throughput analysis of these cultures. And what we then see is what's captured in this little video. You're seeing a yellow tumor cell being attacked by blue T cells and red PBMCs, and eventually turning green, which means the tumor cell has been killed. This is just an example of the imaging that we perform on these cultures. So you will get both the granularity of single cell data at the single cell resolution, but also at a bulk culture level. So, and we can start understanding what's going on a functional level between the controls and the treated samples. So once we have acquired all of these images, we have an amazing software team that can basically convert these images into quantifiable numbers. So you can see the hazy fluorescent images in the background, and these round dots are our detections. So they're what the computer vision pipeline is able to see and quantify and extract in terms of both the XYZ position of the dot in space over time, as well as the number. So quantifying this is done through our pipeline called Einstein ex vivo cell interaction mapping with spatial temporal image analysis network. The images are taken, deconvoluted for each channel, for each color, denoising, go on, background subtraction, and then the images are segmented, turned into those little dots, into those little spots that I showed you earlier. And again, a variety of different metrics. I think we're having over 100 metrics can be extracted and quantified using this method. So some of the most obvious things we can look at is how are the immune cells infiltrating into the 3D microtumor over time? And how does this infiltration change with response to treatment, for example? Other metrics that we can look at with this higher time-lapse, higher resolution and time-lapse is cell migration. So following a spot through time, through space, enables us to quantify how quickly they're migrating and how quickly their migratory patterns can change in response to treatment. We can obviously look at viability. So is a cell alive, dead, or dying? And again, how does that change throughout time with the addition of different treatment regimens? How does the bulk of the culture overall look in terms of volume, diameter, area, and how does that change with treatment? So far with PEAR-GLIO, we've only been in this optimization phase where we're really just gathering data and testing the platform. But the idea will be to run a further 40 patients and linking their outcome, their clinical outcome, to the responses that we're seeing in our assay through this variety of different metrics and seeing if we can create a predictive model. So this is exactly what we have done in another cancer, in triple-negative breast cancer. We have a preprint of our paper out. The link is down here if you guys are interested. So this is what we're trying to get to with PEAR-GLIO as well. The idea behind this trial is a trial in neoadjuvant triple-negative breast cancer. So these are patients that are having their surgery, and we're taking an extra biopsy of their from their breast of their triple-negative breast cancer. And then the patient go on to receiving neoadjuvant therapy, which in breast cancer is a combination of 4 chemotherapies, 3 or 4 chemotherapies, plus the addition of an immunotherapy in some cases. So we run our test, the patient goes on to getting treatment as the standard of care, and then we compare the results of the test to the clinical response of the patient. And we looked at over 40 individual metrics, but even selecting a single metric, which is this normalized delta, we had an accuracy, we were 83% accurate at predicting patient outcome. And this is a really, really good result, especially for triple-negative breast cancer, where response prediction is very tricky. I will say the caveat is that we've only tested 12 patients so far in this preprint, but we have 24 in total that we are waiting for to collect outcomes for. So this number is likely to increase when we add more patients, and is also likely to increase if we combine multiple metrics. Again, this is a single metric that we looked at here, but keep tuned. We'll post more about our trial once we get those outcomes. And so to conclude this part of the talk, we have a number of other clinical trials also running where we're trying to validate the platform and our technology by following the patient and seeing what their outcome is in the clinic and seeing how that correlates to the results that we're getting in our test ex vivo. Not to mention triple-negative breast cancer, our kidney cancer trial, PEAR-GLIO, which is our brain cancer trial, and then we work on a lot of other cancers still at the R&D stage. Last but not least, BPIE. So this was an addition for PEAR-GLIO trial specifically. And so we met with a lot of patients and carers and also clinicians to understand what the perception around functional precision medicine is, and how that changes after explanation, and how we can use, patients' insights and carers' insights and clinicians' insight into informing our precision medicine test development. So here on the left, you have it's a word cloud for what words sprang to mind when we asked what functional precision medicine was before the session. And there's a lot of, understandably, words around not knowing what it is, it being incomprehensible. When we asked the same question at the end of the session, we got much more optimistic, exciting, and much more interesting mix of words. Some of the things that we covered in these sessions were, how would the test help you make decisions around treatment? What type of data would you want to see or would you need to see in order to be more comfortable in your treatment decision making and in using this test? When would it be the best time for a clinician to approach you? And are there any other tests or other data that would be worth us providing you to increase the likelihood of you using the test or trusting the test results? So with that, that's all I have for you. Thank you very much. I'll be very happy to take your questions. And we are based in West London.
Thank you so much, Ellie, for such a wonderful and really insightful talk, and I've learned so much from you. I'm going to open up to the audience if they've got any questions. If not, I've got loads of questions myself. So you can either use the chat function or raise your hand, and then I can allow you to
So we've got one question in the chat, which would be, I would be interested to know how long the viability lasts in glioma cells.
Yeah, so we have some data around that. I can go back to the slide. So we have only tested actually up to day 7. So this is your day 7 images. You can see the green cells being alive and the red cells being dead. So by day 7, we're definitely still retaining a lot of the viability. We've never tested beyond this duration simply because we want our test to be clinically applicable. So we want the turnaround time to be relatively short so that, the clinician can receive their test results and then go on to prescribing treatment. But I mean, just looking at these images, I would assume you could keep them going for a lot longer if you wanted to.
Thank you. So we've just had another question pop in. So it says, how do centers transport tissue to you? Is this frozen? And also to acknowledge it was a great talk.
Thank you. No, so we use fresh samples for all of this because we want to culture the cells live. It's important that they're transported to us live, so not fixed, not frozen, but live. We get them shipped at 4 degrees in tissue transport media. And then when we receive them, we immediately start processing them. And from there onwards, they're kept at 37 degrees. So we have done a bit of work on optimizing cryopreservation so that we can, freeze some of the samples and then viably bring it back up to life again later on. But for the purpose of the assay, and again, in order to fit in with that clinical timeline of being quite quick, we do everything live.
Okay, so I've had another question, beat me to my question. So to what extent is the addition of immune cells pre-activated to the culture's representative for tumor infiltrates in glioma tissue, since these tumors in situ are very immune suppressed?
Yeah, that's a great question. So there's a lot of, even internally, a lot of debate on whether we should have the immune cells there for GBM models specifically. We can very easily take them out. It doesn't add anything. I think one of the purposes of having them there is if we want to start testing more experimental drugs. So we have discussed in the past Pembro, but even one of our own targets, so VISIC-4, one of the targets that we have identified, is very differentially expressed in the tumor versus normal brain tumors. So we're considering potentially, could we use this as a potential target, as a potential drug in brain tumors as well? We wouldn't be able to get over the kind of blood-brain barrier issue with the delivery of the drug to the actual microtumor. But if the immune cells were primed and had a way to traffic to the brain, which we know they do from other cancers, then I still think there would be value in having the immune cells in the culture, because you could see whether the inherent patient biology did enable treatment with these immune checkpoints or not. So in the cancers where the immune cells are present, unable to traffic, just because you have the immune cells there, it doesn't mean that you're going to respond to an immune checkpoint inhibitors. So I would assume it's a similar thing in the brain. If the immune cells were able to be primed by a drug to be more reactive and to traffic to the brain and to traffic to the tumor, then potentially those are the patients that would be more eligible for these kind of treatments. Whereas, if you're not able to prime those cells to begin with, then it doesn't really matter whether you have them there or not, or whether you had the drug or not. So that was a bit convoluted, and I don't know if I really answered your question, but I think there's still an interesting use of having an understanding of whether someone's immune cells from the blood could be reprogrammed to go to traffic to the tumor.
Thank you, that's really helpful. I do think you answered the question. I will give an opportunity for those who are in the audience that want to unmute if you want to ask a question. In particular, if anyone has put something in the chat and they want to ask themselves. Okay, so Thomas, feel free to unmute.
Yeah, thanks. I have two questions, actually. First question, you may have answered it. I apologize if I missed it. What's the turnaround time from processing to full results given to the treating clinician?
Yeah, so it's meant to be like 5 to 7 days. So the assay itself is 5 days, and then a couple of days for data processing and, handing it back.
And presumably in the context of glioblastoma, these results wouldn't sway you away from giving standard radiotherapy, chemotherapy as a first-line treatment. So in that case, how applicable are the results you get on that primary tumor to the tumors at relapse, given they've been heavily treated with radiotherapy, temozolomide, etc.?
So the radiotherapy one is a really interesting question, because actually using this model, we can't really assess response to radiotherapy. There's a couple of other companies, I know, like Matt has linked the papers a few times, of other ways that we could look the genetic signatures to determine whether these patients are likely to respond to radiotherapy or not. So yeah, unfortunately, that's a big limitation of this, is that we can only assess.
And have you got any, you may not, because you've got loads of numbers, but have you got any patients where you've done this on their tumor when they first have a resection, and then you've looked at it again when they have re-resection?
We haven't, but I mean, we'd love to. So again, with PEAR-GLIO, I think we're a little bit earlier in the optimization, but there's no reason, with other cancers as well, there's no reason why we wouldn't be able to re-biopsy if that's something that we could do. That longitudinal data would be amazing. And especially in our metastatic trials, actually, something that we ask the oncologist is usually to tell us what the previous line of treatment of the patient was, if they have had one, because oftentimes we can test that treatment and seeing whether, whether there is actually resistance, whether we're actually to detect that resistance to the previous line. So these cases are very valuable, very rare, and therefore it's very hard to run a full validation. But if we could, that would be, very good. That would be amazing.
Thank you. Great talk as well. Thank you.
Thank you.
Thank you. That really moves nicely over to Charles Davis' question, which says, in the Lancet Oncology this month, Strip says metastases are a different brain tumor. Is it possible in the future to compare primary and brain mets possibly by storage?
Yes, if we can access the brain mets and sample them, yes. We know from other, well, Matt is going to give a whole 20-minute talk about brain mets. So, you know, it's a lot more than me. But yeah, I mean, anything that we can biopsy, that we can get physical
think-
live tissue from, we can test.
I think it's not something we've addressed, but it would be very. We currently take metastatic tissue from breast and kidney cancer in our PEAR-MET and PEAR-TREE2 studies. So, in a sense, if you had brain metastases from breast cancer, we'd be reasonably optimistic that we'd be able to grow it, and therefore one could use our platform in theory, assuming it's validated. Yeah, in theory, the technology lends itself equally well to trying to predict treatment response in brain metastases as it does in primary brain tumors. I will just stress that in reality, it is unlikely we're ever going to get enough brain metastases from each of the tumor subtypes to really make that big enough that anyone would feel comfortable in saying we had predictive ability. Although you could say that if we had 100 patients with metastatic kidney cancer and we could predict response well in those, and some of them were brain mets, which difference does it make? The other issue is we don't consider the blood-brain barrier, so we might well end up predicting drugs that should work well but don't get through the blood-brain barrier.
Thank you for that explanation. I've got another question in the chat. I will just ask Joshua, do you want to unmute yourself and ask, or would you like me to read out your question? Okay, I will read. So you wanted to know that how much of your models are able to share with other research groups. Oh, so you can unmute. So if there's anyone else that wants to ask a question, otherwise I do have one. Okay, Joshua.
Hey, sorry about that. I was just at a busy office. Yeah, thank you so much for the talk. So I'm currently based at Imperial, working on the development of a novel local bioelectronic implant for glioblastoma, and currently testing on basic 2D and 3D in vitro models using patient-derived cells. I was just wondering whether you were able to share any of the protocols or any of the samples you had in the models you presented as a form of collaboration so we could test further on this, or is it something you're restricting for the clinical trial setting in the data you presented?
Yeah, dropping me an email. We'll see what we can do. No, we like to collaborate, like where possible. So some of our protocols are proprietary for obvious reasons, but you know, there's no reason why we cannot set up collaboration agreement or see, you know, where we can be of help. I mean, a lot of the 3D cell culture work that we do is not particularly like revolutionary. So it's very basic 3D cell culture with hydrogels. So, but yeah, we'd be more than happy to share. I think a lot more of the IP is around, you know, the end-to-end workflow and our dissociation method, maybe. But like, yeah, we can have a chat.
Excellent. Yeah, I'll be sure to follow up with email. Thanks so much for that. That's a great talk.
Absolutely.
Thank you.
Yeah, no problem.
Thank you. So we've just had another question from Martine. So how do you rank drug efficacy when comparing multiple drugs on a specific samples? How do you relate the required drug concentration ex vivo to what is reached in the tumor in patient?
Okay, so a couple of points there. So it's an efficacy test, so not a safety test. We will never be able to kind of correlate the doses and say that they're exactly matching the regimens of the ones that the patient will get in the clinic. So the way we do this experimentally is a lot of work goes into optimizing drug doses first in cell lines and then on patient samples. And then for combination therapies, we look at synergy and antagonism. It's like a complex kind of matrix. There's a model, it's called LIST model. It's very well documented in the literature, where we kind of look at what doses these two compounds are acting synergistically, or are they just additive, or are they working against each other, which never happens because these are usually approved combos. So a lot of the work that goes into standardizing compound doses, it's not showcased here, but yeah, we have a whole team that works on that. And so we look at the doses and the mechanism of action of the drugs first as monotherapies and then in combination, and whether each of the drug is retaining their mechanism of action when we combine them. So it's a lot of dose responses, looking at AUCs, so areas under the curve, and seeing the patients that are meant to respond have are more sensitive or less sensitive to the drug, and how that changes the AUC of these curves. For purely cytotoxic, that works great. For drugs that are not directly cytotoxic, we need to come in with a lot of other assays to validate that the drugs are working, like Western blots, phosphorylation of proteins, immunofluorescence, you name it. So yeah, it's not as straightforward, but the point is that whatever we put in the test should be a dose that works on the patients that it's meant to work on, not so much that we can use that dose to inform what the patient is going to get in the clinic. That's not something that that anyone has been able to do, I don't think. Yeah.
Thank you. Does anyone else in the audience want to unmute and ask a question, or can I ask my question? I will give 1 second to see if anyone's going to put their hands up. Otherwise, okay, so Ellie, I've got a double-barreled question. So what drugs are you currently testing for brain tumor patients? And then if, in theory, this worked, could patients and clinicians ask you which drugs they wanted you to test?
So this is the current panel for PEAR-GLIO. Yes to the second question, with a big caveat that, as I mentioned, there's a lot of work that goes into optimizing the drugs before we put them into the test. So if it is a drug that we have already worked on in another cancer and that we're confident in what doses we can use in the test, and that we're confident that the drug works well in our hands, then yeah, I think the idea going forward could even be that we provide on a test requisition form, for example, a list of the drugs that we feel confident testing, and then the oncologist could pick or rank them based on what they want tested on that specific for that specific patient. So short answer, yes, in theory we can test anything and everything. Long answer, as long as we have tested and we know that it works, then we can test anything.
Thank you. And then I think the other question I had was, obviously you've got more experience with breast and kidney samples. Understandably, yes, it's easier to acquire than brain. How much shared experience is there across the tumor types, and does it actually get any easier?
So the overall workflow gets easier every time to optimize, because I think we've standardized these steps of what we need to do. How do we identify 3D cell culture methods? How do we characterize the cells transcriptionally? So I think the end-to-end workflow becomes a lot easier to standardize. I think when we did it the first time with triple-negative breast cancer, it took us 2 or 3 years, and we ended up changing the system completely between the different cohorts of the clinical trial the first time we run it versus the second time. Kidney cancer, we started in 2020, end of 2022, and by the end of 2023, we had a model, so it took 1 year. And now with brain cancer, we've only tested 12 patients within 3 months, and we already have a sort of initial prototype. So it's getting faster and faster every time. There's a lot of the biology that can be translated across, a lot of the steps in the microscopy and the computer vision analysis that can be translated across. The main steps that change are your dissociation and the drugs that you're testing. So those are the two things that take the longest.
Brilliant. Does anyone else have any other questions? I'm aware that I haven't actually let you breathe between questions earlier. I've just been firing them at you. We've had a lot of interest. Does anyone have any final questions, or can I ask one more question? So I think mine's more from a patient point of view, or even just a pathway. How would you say there's been a lot of like challenges from patients like opting in to give their sample, seeing that it's all fresh, or clinicians opting in to do it? And where do you think, is there a bit of like, have you seen a bit of like resistance due to people being worried about the fresh tissue pathway element of it being like couriered between hospitals to your home site? And what barriers have you found?
Great question. So in GBM, I don't think we've had any resistance because, again, the patients are undergoing standard of care operation anyway, so they're just almost donating their excess tissue. So I think we've seen in PEAR-GLIO from our PPIE sessions as well, there's a lot of interest and, very minimal kind of disruption to the clinical pathway of those patients as is. In triple-negative breast cancer, which is the trial that we've published, we actually had a 55% consent rate, which is very, very, very good, considering that these patients actually have to go in for an additional mandatory biopsy in order to be able to take part to the trial. And also they get no benefit in taking part to the trial anyway, because they're not going to see the results of the test. So if you think that like over half of patients were willing to undergo other 2 biopsies that they didn't need to, and then it's not going to affect their treatment pathway. And this is just for for development of a tool. I think, yeah, patients are very, very happy to get involved in research that is potentially going to be benefiting other patients in the future. So we've been very fortunate and the reception has been very good. For the clinician side of things, I think it comes down more to just having a good relationship with the CI, with the hospitals, and making it very easy for them to set up the extra visit, get in touch with the radiologist. And it's usually the radiologist, the one that actually needs to be on board with wanting to get the extra sample and give us what we need, rather than the treating oncologist or the patient. So.
Thank you. You've done an amazing talk and answered all our questions. And as I said, I didn't really give you a chance to breathe between questions because they were just coming up so quickly. So thank you once again. And I think the whole audience just wants to thank you as well. So thank you. But for those who are still signed in, we do have our next Horizons in Neuro-oncology on the 18th of March at 5 o'clock, where we welcome Helen Bulbeck from Brain Trust. And again, please sign up using Eventbrite, and it was put in the chat at the start, and Matt has just nicely read my mind and put it back in the chat. So hope to see you all there again. And as we said, we've recorded this session and it will be made available on our YouTube channel, hopefully in the next week or so. So thank you.